Welcome to ANA Investigates, a monthly podcast featuring the latest in neurology and neuroscience, brought to you by the American Neurological Association. I'm your host, Adeline Goss. Late last month, the journal Science published an investigative report that accused a much-cited publication in Alzheimer's disease of fabricating scientific images. The study under scrutiny was published in Nature in 2006 by Sylvain Lesné, Karen Ash, and colleagues. It claimed that an amyloid oligomer called amyloid beta star 56 impaired memory in mice. The recent Science report found potential fabrication in the images used in that paper and concluded that this potential fabrication threatens the amyloid hypothesis in Alzheimer's disease. Here at ANA Investigates, we decided we needed to ask the experts about that. Our guest today is Dr. Rich O'Brien, Alzheimer's researcher and chair of the Department of Neurology at Duke University. He was interviewed by Dr. Erik Roberson, Alzheimer's researcher, endowed professor of neurology, and director of the Alzheimer's Disease Center at the University of Alabama at Birmingham. Their conversation will help us understand the impact of the recent investigative report on the field of Alzheimer's research. Here's Erik.
Welcome, Rich, and thanks for joining us today.
My pleasure.
So this 2006 paper under investigation last month by the journal Science was about amyloid beta oligomers. And maybe for starters, we should define some terms. So what are the differences between amyloid, amyloid beta, and amyloid oligomers?
So amyloid, as you know from medical school, is the description of a birefringent compound, which is a ways that peptides aggregate when they start precipitating. It can be a variety. Transthyretin is another one. But the amyloid in the brain in Alzheimer's disease is a specific 40 to 42 amino acid peptide derived from a very large protein called the amyloid precursor protein. Now, in 2002, Dr. Dennis Selkoe described the first oligomer paper, which was, I think, important. He said amyloid doesn't just exist in the brain as 40 and 42 amino acids, but it aggregates into soluble groups anywhere from 2 to 4 to 8 to 12 mers. And at that point, Dr. Selkoe postulated that the oligomers might be the uniquely toxic version of the amyloid peptide.
So what was the key finding of this 2006 Nature paper that is now being accused of fraud?
So I'm not sure there was a key finding because the idea of amyloid oligomers was already in the field. What this paper said is that there was a particular form, 56 kilodalton, so it was a fairly large oligomer, and that this was the uniquely toxic aggregation of amyloid monomers.
So what are the allegations of fraudulent data manipulation, and how do they affect the findings of the paper?
Yeah, so the accusations are that some of the gels used were over photoshopped. In other words, contrast and brightness settings were used to make certain bands that may not have been consistent with the hypothesis disappear, and that in other places it looked like bands and gels had been cut out of one gel and plopped into another gel, and that band had been used in several different images in the same paper. None of the findings directly said the results weren't valid, but the presence of manipulated images just really calls into question the entire bit of investigation by, kind of guilt by association.
How do these allegations about this particular paper about A beta star 56 affect the support for the overall hypothesis that amyloid oligomers contribute to Alzheimer's disease?
Well, I'm not sure the field really believed that much in the oligomers. I think some people did, but the idea that amyloid, the 40 or 42 amino acid peptide, plays an important role in developing Alzheimer's disease, it's a hard to ignore hypothesis because of two things. First of all, there it is in the brain pathologic findings of people who die from what is called Alzheimer's disease. And it's been known for 100 years that it's there. And second, there are certain very rare autosomal dominant forms of Alzheimer's disease that produce this A beta monomer that accumulates in the brains of patients with the disease.
One of the most explosive aspects of the Science Investigative paper was a suggestion that much of the work in the Alzheimer's field over the last 16 years since the paper came out was based on these findings. And that claim has received a lot of pushback from researchers in the field. How important do you think this paper was to the way that Alzheimer's research has evolved since 2006?
So I read the paper before I started getting texts and emails about it. Actually, I think I listened to it on a podcast, actually, the Science podcast. There you go. And I thought, as I was listening to it, this is crazy. This person seems to think this idea dominated the field and changed clinical trials and the like. And nothing could be further from the truth. It was an interesting idea, but hardly anything that, grabbed the field and changed everybody's direction of their research.
So not something that is fundamentally the driver of any of the clinical trials that have been done since then.
It was a little extra sauce, but it wasn't fundamental. I mean, I believe not this particular paper, but the entire field of oligomers, and there were a bunch of different proposed ones. I think that led the companies to make sure their antibodies soaked up oligomers when they used it, but it would also soak up the monomers. So I think its actual impact was close to zero. No matter what the form of A beta was that was toxic in the brains of patients with this disease, these therapeutic trials seem to take care of them all.
Thanks, Rich. How has scientific publishing changed since 2006? Do you think that this kind of data manipulation that's alleged in this case could occur in papers today?
So, I'm sure you know the work of John Ioannidis at Stanford, and I've followed that over the years. He's somebody who's pointed out that bias in biomedical research is an important issue, and I tend to agree with him. And a lot of the research that is hard to reproduce is probably subject to the bias of the investigator rather than any outright fraudulent attempts of the investigator to put one over on somebody. I think the journals have become more sophisticated at analyzing figures. I don't know that for sure, but the technique that the investigative reporters used to see that these gels had been manipulated just involved really dialing up the contrast and the brightness levels. That's fairly basic stuff, and you'd think there'd be some software that could examine images and find that out. And I suspect that's been put into effect since that time. But if you're saying to me, if somebody's really determined, I don't think there's a way to pick that up. And I think there just has to be a way for the field to correct itself as time goes along. And as I said, I don't think people are bowled away by ideas till they've been reproduced by an independent lab.
So this accusation has received a lot of discussion in the public sphere, and that discussion has certainly the potential to increase general distrust of science and medicine. What can we do to minimize that damage and help maintain or restore that trust?
So I think that's a really important issue. And I think the investigators in this field need to realize that what they're doing is a public trust and that their research is funded in large part by the taxpayers of America. And they have to keep that in mind. I think that there are some things that are still a little surprising about animal-based and laboratory-based research, that there's not more of an emphasis on electronic lab notebooks, which all the pharma companies use as a way of making sure there's no manipulation of the data, that true double blinding of research isn't that common, and that there's no such person as a research coordinator like there is in clinical trials, where the research coordinator, one of their missions is to make sure all the research is happening according to protocol. I've worked in labs for years where there are plenty of technicians, and I've wondered why they also don't have more of a role like in the clinical world with research coordinators. But I would say to the general public that the field is advancing. It seems to be taking forever because it's just a really, really difficult problem. And that studies like this that pop up now and then really don't affect the field that much. And that the field is an incredibly self-correcting enterprise because everyone wants to get this right. This is a really big problem.
How do you think this report may affect Alzheimer's research going forward, especially for junior researchers?
Yeah, well, I think so far, over the last 10 years, there's been a real big shift away from amyloid, which I think is appropriate. I also think it's appropriate to finally put to rest the idea that amyloid is the toxic protein in Alzheimer's disease rather than a byproduct of a metabolic disorder. And there's likely to be more and more emphasis on apolipoprotein E research. As you may know, apolipoprotein E and its association with Alzheimer's disease was discovered by my predecessor, Alan Roses at Duke, and its importance has only risen over time. There was a publication by the Banner Institute last year, which showed between ApoE22 and ApoE44, there's over 100-fold increase in risk, that almost the entire genetic spectrum of risk from Alzheimer's disease can be explained by ApoE. And there just needs to be more emphasis on what ApoE is doing in the brain under normal and pathologic circumstances. And I think young investigators should take that on rather than the amyloid hypothesis. But there's a problem in the field, which is the mouse model of Alzheimer's disease is fairly unimpressive, and it's not robust, and findings don't translate. And I think NIH understands that with their Model AD program, where they're trying to produce a more robust model of Alzheimer's disease. And I think that's a very good initiative.
Rich, thanks so much for your time today and helping us think through the implications of this interesting and timely news story for Alzheimer's disease research.
Sure, Eric, my pleasure. Thank you.
That was Dr. Rich O'Brien, chair of the Department of Neurology at Duke University, interviewed by Dr. Erik Roberson, endowed professor of neurology and director of the Alzheimer's Disease Center at the University of Alabama at Birmingham. To find a link to the science article discussed in this podcast, you can visit our website, myana.org. For ANA Investigates, I'm Adeline Goss.
This has been an episode of ANA Investigates, the official podcast of the American Neurological Association, and an ANA Education Innovation Subcommittee production. Thanks for joining us. To learn more about today's program and to find out about the benefits available to ANA members, including access to an array of educational programs and resources, please visit myana.org. Dr. Adeline Goss is our host and the executive producer of the show. Episode producers are members of the ANA Education Innovation Subcommittee, chaired by Dr. Megan Ritchie. This is your logistics coordinator, Jen Hurley. Copyright, the American Neurological Association.